One type of major membrane receptor is the ligand-gated ion channel, also known as the ion channel-linked receptors. Common place to find these ion channels are electrically excitable cells like neurons, and the time scale for cellular effect is in milliseconds. Have you guys ever considered how our nervous system, like our neurons and our brain, can interact so quickly? Well, the reason is the ligand-gated ion channels. These ion channels react quickly to the binding of the ligands, so they're commonly found in the cells that need to react quickly to the stimulus. These are transmembrane ion channels that open and close in response to the binding of a chemical messenger, like a ligand, on the outer side. When we talk about the transmembrane ion channels, what we really mean is that they are embedded in the membrane, or are also called as the integral proteins, having a hole through which the ions can move in or out. Let us say this is our ion channel, which has a specific allosteric site to which a specific ligand can attach. This channel at the particular movement is closed. And here we have intracellular environment, and this is our extracellular environment. The shape of the ligand is complementary to the shape of the ion channel and fits right in there, so specifically known as the lock and key model, or more updated is the induced fit model. When the ligand binds, it will actually cause the opening of the channel. The ligand will bind to the allosteric site, means it will be away from the actual ion channels, but when it binds, it will control the opening and closing of the ion channels by altering the protein conformation of the entire membrane. So when it binds, the ion permeability of the entire plasma membrane can quickly change. So once this channel is opened, it will let ions like chloride, sodium, potassium, and calcium to move through the ion channels. When the ions are moving in and out of this will cause change in the electrical properties of the cell. By explaining briefly converting the extracellular ligand signal into the intracellular electrical signal. It is very rare that the allosteric sites are present intracellularly. Two misconceptions must be cleared at this moment. Ligand-gated ion channels are easily confused with two types of ion channels. They are not the same as voltage-gated ion channels. Voltage-gated ion channels rely on the difference in the membrane potential. They only depend on it, but they actually respond by binding to the ligand. The second one is what we call the stretch-activated ion channels. As the name indicates, stretch-activated ion channels depend on the deformation of the cell membrane or the cell membrane stretching, that is being pushed or being stressed. A classic example of receptor linked directly to an ion channel is the nicotinic acetylcholine receptor. Here is our cytoplasm where other organelles like Golgi bodies are present, and above is the synaptic cleft as the extracellular membrane. The nicotinic receptors is composed of 5 subunits and it functions as a ligand-gated ion channel. Currently, it is closed. Binding of 2 acetylcholine molecules results in a conformational change. This allows the entry of the sodium ions, causing the depolarization of the effector cells.